All right, let's dive right in. Today, we're going deep inside our own cells to see how they read the genetic blueprint of life itself. It's a process called transcription, and, it's happening in your body trillions of times every single second. So it all starts with this one question that's kind of mind-boggling when you think about it. You've got trillions of cells, and they all had the exact same DNA. So how does one little group of cells know it's supposed to build a beating heart, while another group knows to become a brain? Well, the secret starts right here. You see, the cell is smart. It doesn't take its priceless master blueprint, the DNA, out onto the chaotic, messy construction site of the cell. No way. Instead, it makes a temporary working copy, and that copy is called RNA. And this whole copying process, that's transcription. It is the absolute first step in bringing our genes to life. So our first problem is all about security. That master blueprint, our DNA, it has to be protected at all costs. I mean, think about it. Our DNA has the instructions for everything, building proteins, fighting off a cold, even thinking thoughts. The last thing you want is to risk damaging it. So it's kept locked away, safe and sound, inside a little cellular vault we call the nucleus. But okay, if it's locked up, how do you read it? You need a messenger. Okay, so that leads us to the next big hurdle. Our genetic blueprint is just massive. We're talking over 3 billion letters. If you just need the instructions for one little protein, how on earth do you find the right page to start copying? And nature's solution? It's pretty brilliant, actually. Right before each gene, there's a special sequence of DNA letters called a promoter. You can just think of it as a giant flashing neon sign that says, "Hey, start copying right here." It's the landmark that all the cell's machinery is looking for. Of course, a sign is no good if nobody can read it, right? That's where transcription factors come in. These guys are specialized proteins. They're like the expert assembly crew. Their one and only job is to scan the DNA, find that big start here sign, and get the whole work site ready for the main event. And that brings us to the star of the show, RNA polymerase. This is the master copier. It's this powerful enzyme that, once the assembly crew guides it into place, will literally move along the DNA strand and build that RNA copy, one letter at a time. And here, we can actually see it all happen. So first, a key part of that assembly crew spots a specific start here signal in the promoter. This is like the first worker showing up on the site, planting a flag on the exact spot to begin. Now, once that first piece is locked in, it's like a beacon for the rest of the crew. More and more transcription factors show up, they bind to each other, they bind to the DNA, and they start building the stable platform right at the start of the gene. And then, the moment we've been waiting for, the master copier itself, RNA polymerase II, gets recruited to the platform. This whole beautiful structure is called the pre-initiation complex. The engine's in place, the crew's ready, and the blueprint is open to the right page. We're ready to go. So the machinery is assembled, everything's ready to rock, and now the actual copying can begin. This happens in 2 main stages: elongation, which is making the copy, and termination. You guessed it, where it stops. The RNA polymerase just kicks into gear. It chugs along the DNA strand, unzipping it as it goes. It reads one side of the DNA and builds a perfect complementary RNA strand. This process of just making the copy longer and longer is called elongation. It just keeps on going, adding one letter after another. But obviously, this copying can't go on forever. Just like it needed a big start sign, the machinery needs a stop sign to know when the gene's instructions are finished. So how does it know when to let go? Well, scientists have a couple of really cool ideas about this. On one hand, you've got the allosteric model. The idea here is that once the polymerase passes the end of the gene, it changes shape, gets unstable, and just falls off. But the second idea, the torpedo model, is way more dramatic. In this version, after the main RNA copy is cut free, a separate enzyme latches onto the little leftover bit, races down the DNA track like a torpedo, and just slams into the polymerase, knocking it clean off. Now, you might be thinking, okay, we're done. We've got our RNA copy. But in complex creatures like us, what comes off the production line is more of a rough draft. It needs some serious editing and proofreading before it can be sent out of the nucleus. So this first copy is called pre-mRNA. And if you look closely, you'll see it has 2 different kinds of information. The parts we actually need, the useful instructions, are called exons. But scattered in between them are these junk sections called introns. It's kind of like finding a bunch of random rambling notes in the middle of a recipe. To turn this rough draft into a final, usable blueprint, 3 things gotta happen. First, a special protective cap gets added to the front. Think of it like a little hard hat. Second, a long tail of repeating letters is added to the back, which helps keep the whole thing stable. And third, and this is the most important part, all those useless introns have to be cut out.
And this process of removing the introns is called splicing. And it is just incredible. The intron is bent into this amazing loop that scientists call a lariat, like a genetic lasso. This loop is then snipped out, and the 2 useful exons on either side are stitched together perfectly, without a single letter out of place. It's unbelievably precise.
So we found the start, we copied the code, we edited the draft. But it turns out this whole editing process has a secret power. It's a power that totally expands what our genome can do in a way that is just mind-blowing. And that secret is called alternative splicing. This is a total game changer. It means the cell doesn't have to stitch those useful exons together the same way every time. It can choose to skip one or maybe include a different one. It can essentially create multiple, totally different final messages from the exact same rough draft. Check this out. This is an incredible example. It's a single gene called alpha-tropomyosin, but by splicing the rough draft in different ways, your body can make totally distinct versions of the protein for your skeletal muscles, for your smooth muscles, and even several different versions just for your brain, all from one gene. Just think about that for a second. From one single gene, the body creates at least 9 different proteins, each one perfectly tailored for a specific job. This is how our genome is so efficient and so incredibly complex. It's not just about how many genes we have, it's about how cleverly our cells can use them. So from a secure blueprint locked in a vault to customized instructions for every single cell type, transcription is this dynamic, intelligent process that makes us who we are. And it's happening right now, trillions of times over, inside of you. Which just leaves you wondering, what other secrets, what other layers of information are still hiding in the edits of our genetic code?